All right. Good morning, everybody. Welcome to Bernard Snell Hall. I don't think we've been here for a while, except obviously for the Department of Medicine. We were here for our 100-year anniversary, but nice to see everybody. This is a very special event because for quite a long time, I think it's been a few years, at least a decade, we've had this event of combining the CVRI Research Day, kicking it off with Medical Grand Round. So it's a nice partnership between the Department of Medicine and the Cardiovascular Research Institutes. I'm delighted to welcome you all here. Before I hand it over to our conductor of ceremonies this morning, I'd just like to begin by respectfully acknowledging that we are located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to enrich our vibrant community. And so with that, I'm delighted to hand it over to our new CVRI director, Dr. Evangelos Mikalakis.
Thank you, Narmin. One of the main research priorities for the Department of Medicine and CVRI is aging research, and particularly healthy aging research. This kind of priority is strategic because it involves so many disciplines, all the way from preclinical work to clinical and multiple specialties, outcomes, population research, and policy making. So one of the key features or the cornerstones of longevity and healthy aging research is how the environment affects our genes. We are born with a certain set of genes, but we can modify them, perhaps with drugs, but certainly with a lifestyle, epigenetically. So this is what we're going to hear from our guest speaker, Dr. Michael Kobor, from the Department of Medical Genetics at the University of British Columbia. Mike came from Germany and did his PhD at the University of Toronto, and then his postdoctoral fellowship at University of California, Berkeley. And he started a career, and he's actually a CRC, TR1, in what is called social epigenetics. It's an interesting new sort of discipline. He directs, he's also the UBC's President Excellence Chair in Healthy Aging, and directs a Healthy Aging Center in Vancouver, the Leong Center in Vancouver. The relevance to cardiovascular research is obvious, as you will see from some parts of his talk, because we treat patients with heart attacks and heart failure and they survive it, but then they go on to die or suffer from dementia or cancer or other AIDS-related diseases. So how all these AIDS-related diseases could be modified comprehensively by lifestyle or drug is perhaps one of the new frontiers in medicine. A lot of people call that medicine 2. It's a new era in medicine. So Michael is Canada's foremost expert in this field and one of the world's leaders on DNA methylation and socioepigenetics. As a result, he collaborates with PhD discovery scientists, internists, geriatricians, family practice, pediatricians. He's interested in development and so forth. So we are very excited to hear your lecture, Michael, and thank you very much for coming.
Well, thank you so much for having me.
Oh, he also said he's never been in Edmonton
Don't take
before.
my jokes away.
No, he hasn't been in Edmonton before, but the only reason he accepted this is because Dry Settle is German.
Right. So we'll get ready. So thanks so much, Vangelos, for the lovely introduction. It's a real pleasure to be here. I should confess that while I'm from Vancouver, I'm not a Canucks fan, so don't throw rotten eggs at me. But even though my son's one of his best friends is Henrik Sedin's daughter, for those of you who remember. But it's great to be here. And yes, I think in Germany, hockey and Tri-Saddle in particular has sort of captured the imagination, from what I understand from folks back home. So what I'd like to do today is really give you some vignettes on our research on epigenetics and the human life course, focusing on a number of things. So starting off with what we believe is sort of the idea that early life environments matter, that they get under the skin to affect health and behaviors across the lifespan, which we call our society to cell paradigm. We'll then briefly touch on some of the challenges and opportunities in current population epigenetics and clinical epigenetics, which are related to things like tissue and genetics, and using our recent work on Parkinson's disease as a bit of a vignette, an example. Then talk about epigenetic age and the human life course, and then turning this epigenetic age into a bit more of a medical aspect with some in part unpublished work relating to cardiovascular disease and such. So, as Evangelos said, our area of research is really social epigenetics. It's a very simple conceptual framework, and that is that we postulated our experiences, environments that get under the skin, that then affect biological pathways, and in some cases drill all the way down into the nucleus to affect potentially persistently the expression of genes and the chromatin structure. Now, really, the very simple secret that you need to understand if you want to understand epigenetics is that this DNA in the nucleus here is, of course, not occurring like this in its naked form ever. Instead, it's packaged into very complicated structures that are called chromatin. It's wrapped around histone proteins and such. And those modifications really are at the heart of epigenetics. And of course, if you want to have a very comprehensive review of all the different layers that are involved in creating the epigenetic landscape and the mechanisms there, of my former postdoc, Maria Aristizabal, who is now a faculty member at Queen's, really put together a comprehensive review in PNAS. As you can tell, so today we're going to talk exclusively about what you see at the upper left, which is the DNA modifications. These are chemical modifications in form of methylation groups. Does anybody remember chemistry? What's methylation? CH3. So very simple, and it's covalently attached to the DNA. It comes actually in various shape and forms, in part when it's being demethylated, but today we're going to really talk exclusively about the cytosine methylation. There are, of course, also histone chromatin modifications, such as histone variants and histone modifications, acetylation, methylation as well. Then there are non-coding RNAs and RNA modifications. All of those really have a fantastic story to tell. But in the context of population epigenetics and in health and disease, I'd say 99.5% of all studies are really focused on DNA methylation. And in part that is because the tools to measure this quantitatively and in population basis have really evolved very quickly over the last 2 decades or so. But for the students and fellows amongst the crowd, I would encourage you to think about there's a much, much bigger world in this space other than just DNA methylation. Now, there's plenty of evidence from many sort of natural experiments, if you wish, that show that, for example, the nutrition and the lifestyle of the mother can affect the epigenome of the child. Chief of this is studies that were done on the Dutch famine at the end of World War II, where folks, Bas Hagemans in particular, showed that 60 years later you can still find a DNA methylation mark in the blood that's associated with whether you were starved or not during pregnancy. This has sort of exploded, of course, this entire field, talking about things like, nutrition, fetal alcohol exposure and such that all get under the skin of the embryo of the developing baby and potentially last for a long time. What probably captured the imagination more than anything in the space of population epigenetics is work of my good friend and colleague, the seminal work, Michael Meany from McGill University, who initially in a rat paradigm of licking and grooming, but later on in humans also showed that the maternal care, so postnatal, is associated with the epigenome of the child. This, of course, as you can imagine, made the center page of the Globe and Mail and CBC and all sort of things, but really highlighting the fact that these early experiences can get under the skin to affect health and behavior. Now, you probably are familiar with twin studies. They're very often used in sort of human genetics for sort of genetic investigations, but it also turns out, do we have any twins in here? Do you know any twins? Are they very similar if they're identical? Even if they're identical or not, right? So you look at these two identical twins here, you look at, happy baby on the right and screaming baby on the left. It turns out that identical twins, even though they have more or less the same genome at birth, they develop discordant epigenomes during their life course, in particular if they happen to be separated early in life. And in turn, this can then be also associated with different chronic diseases of the life course and aging, including mental health, cardiovascular disease and such, again suggesting that the epigenome operates in addition to the genetics to shape our health and behavior across the lifespan. I'll tell you a little twist though when in 7 minutes, so stay tuned. So in our laboratory, we really look at epigenetics and child health at the sort of interface of nature and nurture, where the nature is the two strands of DNA we get from our parents at birth. And the nurture is the sort of the fertile grounds of our families, of our communities, that ideally we like to bring them together through the lens of epigenetics to best understand how we can give our children a healthy start and prevent importantly diseases across the lifespan through optimizing some of these things, and in particular, of course, the environments, because we can't really change our genes unless on CRISPR. And we do this very much in a sort of developmental lens. So this is a review that my former postdoc, Meg Jones, who is now a faculty just over in Manitoba, put together some years ago, where we postulate that during the human life course, there are, of course, sensitive periods, and one of the primary sensitive periods, of course, is during birth, as we briefly touched upon in early in life. And while our genome is more or less stable, our epigenome potentially can be altered. And so if, for example, a hazardous exposure might happen during one of these sensitive periods, it might alter the trajectory of our epigenome, as you can see in that red line, to then ultimately lead to a higher risk state in adulthood or in older age. If, however, the same exposure happens later in life, it might not actually have such a detrimental effect. Now, of course, I now think some years later, there are more sensitive periods like adolescence and puberty, and potentially also age-related sensitive period. But I think the point here is really that these exposures potentially associate with changes in the epigenome. And my really dear friend and former mentor, the late Clyde Hertzman, who died way too early a decade ago, put this very succinctly, these ideas into a wonderful review. It talks about this sort of biological embedding of early life experiences and the effects of health in adulthood. It's mandatory reading in my laboratory for everybody, but it's really eloquently written. And so we, over the years, have been focusing particularly on one exposure that happens early in life and has profound effects, and that is poverty. So we recently, as part of the social exposome cluster that I was directing at the University of British Columbia, put together a review that led by my wonderful colleague Kim Schmidt that talks about these multi-systems associations of child poverty. And so they go from these sort of early impacts on the child's biology and including DNA methylation. I'll show you some data, but also things like the microbiome and brain structure and so forth, to the more sort of intermediate impacts, developmental outcomes, and most importantly, I think for this crowd here, also the long-term implications for adult health and well-being. Then increased things like increased risk susceptibility to infections, risk for mental health problems, inflammatory phenotypes, increased health risk of chronic diseases, and ultimately decreased productivity, all tracing back to early life child poverty. So we set out some years ago together with psychologists to test whether it is in fact this sort of circuitry between DNA methylation and gene expression that perhaps might be in part responsible for some of these long-term effects. And so what we did in really to assemble a team of wonderful colleagues to allow us to rigorously test these ideas of early life socioeconomic status, but also early life parental stress. So does it matter, whether you live in a family with doggy woof woof, the nuclear family, or family where the parents constantly yell at each other? And so in a series of papers that we published in PNAS, we showed that if in an adult cohort in Vancouver, healthy adults that were basically stratified by their socioeconomic status early in life, so the first 5 years in life, but mixed in adulthood, we found really that there was a biological residue of early life social class. We could see that at a level of gene expression, where basically you see these sort of bioinformatic analysis of genes in the blood that are changed between the two groups. These are not random genes. These are genes that are regulated by pro-inflammatory transcription factors, suggesting that there might be a pro-inflammatory phenotype in folks who grew up in low socioeconomic status. When we then test this by stimulating their innate immune system, the TLR pathways, you can see at the upper right that in fact, folks who grew up in low socioeconomic status have a higher TLR response than those who don't. At a level of cortisol at the bottom of the left, we can also see that these folks have a higher level of cortisol, so they appear to be more stressed, naively speaking. And in a follow-up paper, we showed that there is an association also of this early life socioeconomic status with DNA methylation in the blood as judged by these skewed p-value distributions. So collectively suggesting that there's a biological residue that we can measure. And this is independent of current socioeconomic status that traces back to whether these people grew up in low versus high socioeconomic status. Now, of course, these things you always want to test rigorously and see whether they actually broadly apply or whether we just happen to have findings. So we have been setting out ever since the last decade or so to test across different populations, to just give you some flavor of what we have done. So together with my good friend Tom Boyce and Nikki Bush at the University of California, we looked in an earlier life. So this is an adolescence cohort called PAWS, Peers in a Wellness Study. And it's a different tissue. It's cheek cells, which is much easier to get from children. And it's a more sort of nuanced unpacking of socioeconomic status, including looking at income per dependent, but also parental education, family adversity. And each of those in these sort of more than 200 kids, we can find, again, a skewed p-value distribution. And we look a little bit more closely in these at the bottom. It's called volcano plots. These are individual of these CBG sites that are associated with all of these three variables. What you look in a volcano plot is something that is high up because it's more statistically significant, and it goes away from the middle because it is more biologically relevant. And so you can see that there's overlaps of particular of these CPGs, several hundreds each for these various things. Again, suggesting that in this very different population and in a very different tissue, we actually see these associations. More recently, we have been collaborating with my good friend Tom McDade and Chris Kusawa, both at Northwestern. Actually, Chris just moved to Harvard, right in time for the boycott of Harvard. And looking at Philippines, so they're long-standing collaborators of the CEBU longitudinal study of health and nutrition survey. And again, what we found in a series of papers is the same idea that social and physical environments early in life do associate with DNA methylation, and particularly of inflammatory genes. We're also able to use this information then to calculate an inflammation index based on DNA methylation, which gives you a little bit of the flavor that we'll come back to later. So these are just some really brief examples. We have really expanded this work to look at things like, the family stress. We have worked with Megan Gunnar to look at DNA methylation in children that spent the first 2 years in these awful orphanages in Romania before being adopted to the United States and families. We have looked with Andrea Roberts at Harvard at sperm to find signatures of child abuse in sperm and so forth. You can see this here. And we've also taken this a little bit more into the clinical diagnostic space by looking at more recently at fetal alcohol spectrum disorder and trying to see whether the epigenetics can tell us something about the biology of the disease and or opportunities to diagnose earlier or better. Happy to talk about this later. But I want to move on to really share with you some of the challenges and opportunities. And again, I am German, so we're supposed to be always sort of negative and glass half empty, but I'm actually glass half full kind of person, which is probably why I live in Canada. So one of the big challenges, opportunities, the challenge in epigenetics is that in contrast to your genome, your epigenome is actually different depending on what cell, what tissue you look at. In fact, during development in all of us, the DNA methylation in particular actually helps to differentiate and maintain tissue differentiation and cellular differentiation. So, I bet, and you can see a lot of our work is really focused on behavior and things like that. From living humans, we can only really get blood and cheek cells, and we were interested in the brain. I don't think anybody would give me a piece of their brain. Volunteers, please tell me afterwards. And so we have been long trying to understand how can we really sort of draw information from accessible tissues about tissues that we can't access. And this is one of the things, just to highlight work we've done again with Michael Meany and Gustavo Turecki and McGill, basically showing that even in matched post-mortem samples that we can find, even for something like aging, we can find both concordant and discordant methylation signatures in blood and brain. So we find some signatures that are conserved across brain and blood and others that are not. We have then also created a tool for anyone who's interested in this that was published, is highly cited in Translational Psychiatry, where my former bioinformatician Rachel Edgar, who's now finishing her PhD in Cambridge, basically the set asked, can we make this an accessible way for the community to learn what kind of DNA methylation site might be conserved between brain and blood. And you can see in these volcanoes at the bottom there, these are like the best volcanoes we ever get, because the difference in tissue difference between brain and blood is just so huge. So you see all these blue and red little dots, all these methylation sites that are different. If we compare two brain areas, it's much less so in the middle there. And so you can basically, for all the CPGs that we measure with our tools, you get an information to the extent that they're conserved between brain and blood. Of course, ideally would want to take this even further and look at many, many different tissues, and we're in the process of doing this. The other thing that we also realized, that contrary to what I showed you or told you, that epigenetics, with the identical twins, that it's not really anchored in the genome. It actually turns out it really very much is so. And so we've been part of a series of papers going back to 2014, where basically we ask, or our collaborators ask the question, if we look at variable sites in the DNA methylome, how are they best explained? Are they best explained by the environment, like I probably just told you in the last, 5 slides? Or are they best explained by genetics or by a combination thereof? And without going into too much detail, it turns out that it's really the combination of genetics and environment that best explains variability in the epigenome. There's very little that's environment alone. And so what it means is that our genetic background matters. And this might be in part linked to, you can see on the right there, this idea of methylation QTL. So these are allelic variants that are associated with the methylation of nearby CPGs, depending on the allelic status on the genotype. And so this is a really exciting emerging field. I have a wonderful new student, Eric Navarro, who just has a paper in review, where he created a package called ramen, much in line with favorite food in Vancouver, that basically allows you to sort of operationalize these kind of measures. And so really exciting. We have been actually using this sort of approach in a clinical setting in work that a former student of mine, Sam Schaffner, has done, focusing on Parkinson's disease. And so what Sam realized, and she wrote this, this is from her thesis at the left there, is that Parkinson, it kind of fits all the things that we're interested in, because obviously there are some very strong genetic associations with Parkinson's disease, like LAUC2 and so forth. But there's also a really very well-established gene-environment interaction. So, environment and lifestyle can affect the onset or the severity of Parkinson's disease. And you probably remember, that actually smoking is good for preventing Parkinson's disease. But of course, a lot of the disease happens in the brain. And so what Sam has been really trying to do is to understand, with a focus on alpha-synuclein, which is one of the main players in Parkinson. Actually, you can see on the top there, physically interacts with enzymes that create the DNA methylation landscape, the extent by which genes and environment interact in Parkinson's disease. And initially, she started together with Tiago Terrio and my first PhD student, Julia Schulze-Hendrich, looking at alpha-synuclein overexpression in a cell line, and found that indeed there was an epigenetic sort of signature that pointed towards glutamate signaling and locomotor pathways in that cell line consistent with the disease that she could identify that was caused by the overexpression of the alpha-synuclein. But then she also went together with the colleagues in France. She then looked at this sort of idea of pesticide exposure using a French agricultural cohort of a couple of hundred people and using whole genome DNA methylation and genotype. And what you found is that, again, you look at these volcano plots at the left, that there are some individual CBGs that are associated, particularly in females, with Parkinson's disease. When she then, and I'll point here to the bottom right, when she then asks, to what extent are they best explained by the environment, so the pesticide exposure, or the underlying genetics, so the genotypes, or a combination thereof? Again, she finds very much that they're primarily best explained by genetics and G by E combination, similar to what I showed you from the other cohort. So really highlighting the fact that we need to look at the epigenome in the context of the genome as well, moving forward. Now, having sort of told you a little bit about these opportunities and challenges, I want to move on to talk about epigenetic age. Who has not heard about epigenetic age? Nobody. Okay, so then you can give the lecture, the rest of it. So it turns out the epigenetic age, so everything I've shown you so far is really an epigenetic association. We sort of ask, what kind of environment exposure or health outcome is associated with individual CBGs or potentially pathways like immune pathways. As we briefly touched upon, one of the challenges is that it could be very tissue specific, so you can't necessarily measure something in blood and conclude that it's also happening in the brain or in the kidney or wherever else. And as these tools evolve, it also becomes statistically very complex. So, when we started doing this, we measured, I think, 1,500 CBGs, and now we're measuring 1 million. And so you see that there is statistical issues. So it turns out that there's actually some other ways, in particular the epigenetic aging, that by which we can harness information content of the epigenome. And this really was conceptualized and pioneered by my good friend Steve Horvath when he was still at UCLA. And what Steve has recognized is that as we go through the life course, many of these CBGs change. And what he then did, he just basically grabbed all the publicly available data and used machine learning to basically create a bioinformatic predictor of age based on DNA methylation. And because he used all kinds of tissues, this original predictor is quite tissue agnostic. It's statistically actually very simple, because all you do is you compare your predicted epigenetic age to your actual chronological age. And so, most people, not me, but most people can analyze this kind of data in Excel if they're so inclined. So what does, and we can derive all of this information from the very same tool. This is not to advertise it, but this is the sort of workhorse, which is an Illumina microarray on the left there, which is 8 subjects on there. And we can use that information to do both epigenetic associations and epigenetic aging more, where we can also now predict epigenetic risk scores such as smoking, inflammation, and predict the nature or the composition of underlying cell types in a sample. We're going to touch upon all of this. So what does this look like in actual reality? So we'll show you some data from the CLSA. Does anybody, do you folks know the CLSA, the Canadian Longitudinal Study of Aging? I don't know whether Edwin is actually a site or not. It's a wonderful thing that is led by Paminda Reiner. And we actually created, I think, 1,500 profiles of DNA methylation in the participants of the CLSA. And so what you see in this slide is we basically did an association study on the left, where we asked, are there individual methylation sites that are associated with age? And again, in this volcano, you can see some very statistically significant sites, such as these ELOFL2 CPGs that are associated with age. And this goes both ways. And I put this in for Evangelos to illustrate that some of these methylation sites, as you get older, go up and others go down, as you can see at the bottom there. So really exciting. And then you take that very same information and you compute epigenetic age. And what you can see on the top are 2. So it's the original gangster, the OG, the Horvath clock, and the Hahnemann clock, which is a blood-specific clock. And what you can see here is basically a correlation between your chronological age and your epigenetic age. And you see there's a very good correlation, but also there is some scatter. And then the question, of course, becomes what explains the scatter? And the one thing that is probably the most replicated finding in epigenetic clocks is that females are always epigenetically younger. So what does that mean? Well, again, my former postdoc, Meg Jones, put this really succinctly in a great review in Aging Cell. We published a decade ago now, where what Meg postulated that most of us are in this concordant zone, where basically our predicted epigenetic age more or less corresponds to our chronological age. But then there are some folks, like, for example, Jason, who goes running every day for hours, who might be, I think, maybe 40 chronologically, but 30 epigenetically. So epigenetically younger, and others who maybe drink a little too much or whatnot and are epigenetically older. And so this then can be translated into different health outcomes, where, obviously the folks with epigenetically young have better health across the lifespan than the folks who are epigenetically old. And there are many, many studies that really point to this. Mostly they're kind of figuring out who's epigenetically old, and other than females being epigenetically young, there are fewer studies that focus on who keeps young, although that is rapidly changing. Now it turns out that this clock business is really sort of evolving very, very quickly. And so there are now very large toolkit of epigenetic clocks. So there's the sort of first generation clocks like the Horvath clock and the Hahnemann clock that I just showed you. Those were really trained on chronological age. So to some extent, they really reflect your chronological age. But then people then have started to sort of develop second generation clocks, which were trained on more complex markers, including age-related phenotypes, smoking, and in some case, even time to death. So the Krim clock was trained in part on time to death when the Krim Reaper comes to get you. Really, and in both of those cases, the best practice is to then basically regress the epigenetic age onto chronological age to find what's called epigenetic age acceleration. And then there's the third generation clock, the most recent clocks that are really based, not so much the one trained on chronological age, but in using longitudinal DNA methylation data. And so they really give you a better idea of the sort of pace of aging. And the Dunedin pace of aging is an example that we're going to see a little bit in a few slides. So keep that in mind when you sort of look at clock papers. They really tell you different things, and they can, because they're trained on different variables relating to age, and potentially also on different tissues, of course. We have then gone back to our original question about early life socioeconomic disadvantage, and together again with Greg Miller and Edith Chen, some years ago found that people that grew up in poverty the first 5 years of their life do have a faster accelerated epigenetic age in their monocytes in this case, compared to folks who grew up in high socioeconomic status. And again, this is independent of the current socioeconomic status, again, suggesting that indeed early life poverty gets under the skin to potentially stick with you for the rest of your life. We've also ventured far into more sort of natural experiments. And so together with my friend David Rekoff at Stanford, we are looking actively at the blue zone epigenetics project. And you probably know the blue zones of regions of high longevity. There are at least 4 in the world, including one in Evangelos, where he comes from, Greece, but also Sardinia, Okinawa, and Costa Rica. And we have really focused on primarily in Costa Rica, but also a little bit in Italy and Greece, asking questions of, is there an epigenetic signature of longevity in these blue zones? And my former super talented student, Lisa McEwen, some years ago tested a very simple prediction that people in the blue zones would be epigenetically younger than age-matched people outside of the blue zone because they're healthier. Turns out they're actually not, at least not by the measures of these first-generation clocks that we used at the time. However, what Lisa found, and we have replicated this now, is that people in Costa Rica tend to be almost 7 years younger epigenetically than people that are not in Costa Rica. We still don't understand exactly, is that a question of the training data sets, or is it really something that reflects biology and longevity? So if you have any ideas, I'm happy to discuss. As I mentioned, we cannot just compute epigenetic ages from these. We can also compute things like cell type compositions and various things. And so what Lisa found in this cohort is actually that the folks in Nicoya, which is the high longevity area, compared to their age-matched cousins outside of Nicoya but living in Costa Rica, actually had what we figured out was a younger predicted immune phenotype, in that they really had, the Nicoyans had a higher estimated proportion of naive versus memory T cells. And we're now testing this more directly with more measures, but basically pointing to the fact that longevity and the immune system aging might be tightly connected in these blue zones. We then moved to the Greek and the Italian blue zones, or high longevity region as they're called now. I have to say, this is one of the experiences where my student Hannah Ruth Engelbrecht, she really made lemonade out of lemons, because we could not get any metadata other than age and sex. And so what Hannah Ruth did in a recently published paper, she basically looked at epigenetic signatures with these various clocks between comparing men and women in these high longevity regions. And the key of these high longevity regions is that actually men, contrary to most other societies and most other regions, men are equally healthy to women in older age. Now, what Hannah Ruth found is that actually on the clock, in all of the clocks we tested, the women are still epigenetically younger than the men, sort of not consistent with the fact that they're equally healthy. She also found, she used this methylation information to compute scores. And so what she found is that men still had shorter telomere lengths. Men had higher smoking scores, but the one thing that she found where men and women were equal was serum IL-6, or interleukin-6, which is to some extent the mark of inflammation. And again, pointing to the fact that perhaps some of these longevity and the balance between females and males in this particular study in these two regions could be best explained by immune system function. And again, we're exploring this further within the constraints of trying to get samples out of Icaria and Sardinia is not easy. So if you have any. Now, I want to finish my lecture here by using some vignettes of ongoing studies in part, but also just to give you a flavor of where this could actually go, these epigenetic locks in these predictors. But before I do this, do we have any Kardashian fans in the crowd? Yeah, you? Did you see their epigenetic age? You did. Did you love it? Good. So these are the Kardashians, and not that I, and there's a really, I think they're on TikTok and everything, right? And talking about, and then they're actually using the pace of aging, but they talk about a Horvath and make fun of Horvath. So they don't quite understand. But I think, you can laugh about it, but I think what it really has done, so everybody, a lot of people now know about epigenetic age because of the Kardashians. And then many of you probably have heard of this guy, Brian Johnson, who's infamous, who spends, I think, $2 million a year on keeping young. And part of his regimen includes 80 pills a day, but also the occasional injection of his son's blood, plasma, to keeping young. So you see him here with his son. I think he's 48. So a very interesting example of and he actually has an exclusive. He claims he's the world record holder in being epigenetically young. Although apparently recently got beaten by an 80-year-old woman, so it got really upset. Now these longevity tests now can be over the counter. You can send your spit or your saliva or your cheek cells or your blood to companies to get your epigenetic age, and we can talk about later how valuable this is versus not. We have used this in the sort of more aging space. Again, going back to the CLSA, we have looked at whether some of these epigenetic clocks are associated with frailty. And indeed, if you look at particularly the Grimm age here in this plot at the bottom, you can see that when we adjust for age and sex, only they're very much associated with epigenetic age acceleration. Once we sort of put in other socio-demographic factors, the association is dampened a bit, but still there. So suggesting that people that are more frail age epigenetically more quickly, perhaps not entirely surprisingly. We have also, and this is unpublished, so if you happen to be a reviewer, please be kind to us. We have recently teamed up with, by chance, with two cardiologists in Texas, particularly Parag Jain at Southwestern and Paul Chachia at Baylor. And the idea, so they have this cohort of young adults that have undergone the Fontaine procedure early in life. And you can see it's a very small kind of group, and their controls are basically nurses, and then 10 or 13 Fontaine procedures, and then single surgery controls. You see the, you're probably much more familiar with all the clinical aspects thereof. But the bottom line is that is Fontaine part of the group, they underwent these procedures when they were fairly young, and the single surgery had just one surgery at around the same time. And so we ask, is there in their blood, is there an acceleration of epigenetic age? And as you can see at the top here for both pheno age and Grimm age, if I see it correctly, there was indeed an acceleration. The orange is the Fontaine group, the gray is the controls, and the blue is the single surgery group, suggesting that these, and there's no association with the generation 1 clocks, either Horvath or Hahnemann, but with both the generation 2 clocks, where you can see again the Fontaine group aging more quickly than the control group and the single surgery group. And the same is true at the very right when you look at the need and pace of aging, again, suggesting that, again, it's a very small study and there are some caveats to it. And that we're still, so I'd love to talk to folks in the room more, whether this could be expanded and done perhaps in a more sophisticated and in a bigger way. But we also use then, again, the same information to compute inflammation scores. And what we found, Beryl Zhang, a bioinformatician in my lab, is that there is an immune marker vestige of the Fontaine procedure. So when she computes IL-6 score or CRP score based on this DNA methylation, we again see that the Fontaine folks have a higher level of IL-6 or CRP compared to the controls. And these are fairly large effect sizes at the group level. Of course, you can see that it's not true for each individual. And again, with the single surgery sort of being in the middle. So, happy to discuss this further and get feedback from the experts on what you think that means and how it reflects what you see in the clinic. Now, with all this epigenetic age, we sort of realized that being epigenetically older might not be all that beneficial generally at a group level. The sort of next final frontier is, can we reverse epigenetic age? So some years ago, we teamed up with a fellow who's really passionate about freezing himself. He's cryopreserving himself, but also very passionate about sort of anti-aging. And so this is Greg Fahy in California. And together with Steve Howard, we ran the methylation arrays. And what Greg Fahy had done was a very small trial, a pilot trial called a TRIM trial, which is really aimed at regenerating the thymus, restoring immune function, and mitigating the effects of insulin. So what they did is it was like 10 or 12 middle-aged males that underwent a sort of treatment for a year of recombinant human growth hormone, some testosterone derivative, DHEA, and also metformin. And then we ran these in blood, we ran the methylation arrays and computed epigenetic ages. And you can see that for all of the epigenetic clocks that were tested at the time, the epigenetic age acceleration goes down, especially at 12 months, which was the end of the trial. And it sort of bounces back a little bit at 18 months. And so suggesting that at least to some extent, this could be reversed. I'm still not 100% convinced of it because I still think there could be some underlying cell tissue differences that we don't really capturing. But Greg is now taking this, I think, in a more larger scale trial to see whether he can confirm that. Anecdotally, one of the folks in the trial apparently had gray hair, and then after the trial had brown hair again. So who knows? But just to give you a sense, and perhaps in a bit more in a larger and more rigorous studies, we were part of the calorie trial that was run out of Duke University that was published last year, which is basically a comprehensive assessment of the long-term effects of reducing caloric intake. This was done in healthy adults. It was a randomized trial, and you can see sort of the layout on the right. And again, what we did here, we ran these methylation arrays and then simply asked the questions: how does the epigenetic age over the 3 time points sort of pan out in people that were part of the caloric restriction versus the control group. And again, I point out, these are not overweight people or unhealthy people, these are healthy people. And so in this paper, what we found that if we look at pheno age and grim age, there really was no difference between the two groups over the course of this trial and afterwards. But if you look at the median pace, which is generation 3 clock, indeed, you can see at the bottom here, the two groups kind of separated. Not every single person behaved the same way, but at the group level, again, the caloric restriction appeared to slow the epigenetic pace of aging. So there's some good news. And again, this is now being replicated and perhaps brought into a more clinically relevant cohort. So all that said, we have really tried to, be very collaborative and take a particular look, bring epigenetics to various aspects of clinical and population health. And this is reflected in the new Edwin Leong Center that I have the privilege to be the inaugural director of, where basically what we focus on in our center is the biology of aging, the gerosciences, with the this is based on the ideas that were put forward by Felipe Sierra some years ago, that the biggest risk factor for all age-related diseases is really age. And so understanding the biology of aging is crucial. We do this in a context of society to cell, like the blue zones and other things, but also trying to then understand what are the best ways of prevention and intervention and bringing that unique lens to age-associated diseases. And one of the areas that we're really passionate about, that I think will help us to separate from, all these longevity and aging centers that these middle-aged white male billionaires start, like Altos Labs, is that we're really focused on the social determinants of healthy aging. And again, using an interdisciplinary lens, using a collaborative lens, and community engagement. Most important, bringing our life course perspective. And this sort of brings me to my sort of last slide, which really highlights that we look at aging as a lifelong journey. That basically, if you take your typical middle-aged person, we have all these determinants of current health that can be traced back to either prenatal or early postnatal life that we talked about in this presentation, such as this idea of biological embedding of, socioeconomic status, poverty, stress, and so on. But also a diet and exercise, and to some extent that determines our current health. But then we also have an opportunity to look forward and using this sort of multi-omics integration, wearables, big data, machine learning and such, to predict how a person's health will evolve over the life course. And I think the aging clocks are really a lovely tool. I talk about epigenetic age, but there's also aging clocks based on microbiome, on brain age, and so forth. And in our center, we really focus on health span versus lifespan. So can we, optimize how long people stay healthy? And we also always straddle that fine balance between population and individual health. As you probably see in all these clocks, there are differences between groups, but it doesn't necessarily mean even for the front end that an individual you couldn't just pick one individual easily and say they belong to group A versus group B. And so this is important to keep in mind. It's also important to keep in mind when you think of the take home sort of epigenetic clock kits that the Kardashians and others are peddling. We very much think of this in the context of intervention. So when is the best time to intervene, and what is the best intervention? We sort of look at interventions very broadly. They go from the more typical like nutritional, physical, but also all the way to things like economical intervention. So until very recently, we had an R1 grant that was canceled to look at basically basic income and tax support in the United States in a low-income cohort of adults and look at their aging, but this was canceled. So we'll have to sort of see what we do with this. But I think aging, we look at it as a lifelong journey. There really is a need to collectively and comprehensively characterize phenotypes across the lifespan to best understand how we can keep everyone healthy and living long and happy lives. And so just to finish, hopefully I gave you some idea that flavor that we think the epigenome is really at the nexus between experience environments and phenotypes and health. We need to look at this very much in the development context while being aware of things like tissue specificity. And I do think, and maybe that's just because we look under the lamppost, that the immune system plays a key role in all of this. And so just to acknowledge the wonderful people that work with me, both in the center and in my lab, and really they're doing all the heavy lifting. I just go around and talk big. And I think I mentioned all the wonderful collaborators as much as I could, and funding agencies. Want to also highlight the Tai Hong Fai Charitable Foundation, which allows us to have this Leong Center for Healthy Aging. Thanks so much. Look forward to the questions.
Thank you, Mike.
And while I'm doing this, I just want to show you one more slide. If this was too much, there's an epigenetic shampoo for greasy hair, or you can do epigenetic yoga to open your genes. So,
okay. Any questions from the audience? Giovanni and then Norman. Sorry, Giovanni. Can you hold on? Get the microphone.
I was about this, and we feel we start to really focus on evolution, because we see that can influence the development of the development, and also the decay around party. And there are some that considerations with purpose are really impacted with the along with any of those studies and what we those but
yeah, so it was a little hard to understand once you moved to the, but if I understood correctly,
I think he wants to your comment on how the pollution,
Yes,
the air particles
Yes,
we hear about,
exactly,
he's a respirologist,
yes,
how
yes.
could this affect
Yes,
epigenetic
yes,
aging?
yes. So it's a great question, and we have a grant in to do this. We have been working, you probably know Chris Carlston in Vancouver, my good friend and colleague. So we have been working with Chris. You know, he has this chamber where he puts people in and then pumps in diesel gas and all kinds of stuff. And we have done the association studies, so not the epigenetic age, but the sort of pathways, and found, you know, your typical sort of inflammation pathways. And we also done with Chris, he's done like even the bowel and all that. We have not yet looked carefully at the epigenetic age in those experiments, but we do have also a new faculty member in the center who had she had the title page of Nature for her postdoctoral work in the UK, where she showed that in never smokers that have lung cancers, there's a very sort of strong association with exposure to PM 2.5. And she's now looking, she just got a grant now to look at how that might be related to accelerated epigenetic age in the lungs or in the blood of these people. So stay tuned, it's a fascinating field, indeed. And of course, you know, we actually have if you think of things like wildfires, right? I mean, these things, you know, how do they reflect? Yes, that's true.
Thank you very much for a sitting presentation. Your conceptual comment this issue of plasticity or state readiness and utilities really sexual.
Yeah, that's a fascinating question. And in fact, together with Tom Boyce some years ago, we tried to conceptualize exactly this, because as by now there's this sort of like almost like a pull and tug kind of ball, because you do have in all of us these sort of methylation marks that are being put down from the moment of conception that underpin, you know, tissue generation and tissue maintenance, and so tissue development. And then the question is to what extent, so for example, the environments, you know, how do they come in? You know, very simplistically speaking, even the strongest of environment will never change the methylation profile of a blood cell to be like a brain cell. There will always be a blood cell, there will always be a brain cell. I think this plasticity there is much more nuanced. It might lead to the cell producing a little bit more interleukins or something like that. So we have, I think, you know, if I were to make a guess, and we try to do that at some point, 95% of the sort of methylation, at least, is the sort of developmentally driven tissue kind of methylation. Then maybe 5%, that's where, but I think that's where the interesting 5%, because that's associated with health, with, you know, longevity and so forth. So I can send you the review if you're interested. I think it points it very nicely.
Narmin, and then Jason. Yeah, okay.
Thank you.
You know, if we talk about the glass people, because of course, as a clinician, what I commonly see is patients that are subjected to that lower thought, socioeconomic status, and the results of you know, those changes in that environment. It was difficult to see that graph up there. One thing you didn't touch on too much tonight, and might have been purposeful. We do find, of course, in clinical practice, much easier to get people to take drugs or having change their lifestyle. And so there was a little like 4 minutes there, but I wondered if you could comment on pharmacological aspect of methylation and the withdrawal.
Yeah, I think, you know, our own work, it has been mostly focused on the sort of social environments, which is what is reflected. And you're right, in that mini trial with the TRIM trial, there was metformin in there. We haven't really looked extensively at drugs, but of course, some of these drugs actually might directly influence the methylome, even. So we're currently doing we just got sort of pilot funding to look at Ozempic and how whether it actually does change the methylation profile, and sort of in particular with a focus on aging, of course, as you can imagine. So I think it's it's an important area. So we're also trying to bring this gerosciences into the clinic. There's a new Canadian network called the Clinical Translational Gerosciences Network that I'm a part of. And in part it is really sort of trying to bring the trials. There was recently a paper published in Nature Aging by a Swiss group where they also looked at epigenetic clocks in a sort of combination of pharmacological and lifestyle interventions. And what they found, perhaps not surprisingly, that the combination was really giving the biggest bang for the buck. But yeah, it's certainly an evolving area. Together with Chris Carlson, we had looked at some of the anti-inflammatory compounds. This was with Johnson & Johnson or whatever they call them, how they might change the epigenome in a nasal epigenome in response to again this PM2.5 expo. So it's an area that we're very interested in. I think the field is very interested in. So lots of work to do.
One more question.
Yes, I'm not always serious. Taking a look. How will DNA determine the effect or the affected expression? But would DNA how a person in the medications that were instead of
Yeah, wonderful question. I might take it one step back. When you talk about the chicken and the egg, I thought you would be asking whether it's correlation or causation of everything I showed you, but it's all correlative, of course. But the idea of the genome intersecting with the epigenome, in part, there are a few aspects to it. And one of them is, as I pointed out very, very briefly in passing, that there are certain allelic variants that are associated with methylation at nearby CpGs. So for example, say if you have an AA, the nearby CpG might have 10% methylation. If you have an AG, it might have 30%. If you have a GG, it might be 70%. So that is part of the story. When it comes to gene, and so you probably also heard of eQTLs. These are expression QTLs. They're the MQTLs. They're somehow overlapping. The methylation itself, we have shown it a long time ago, is not always correlated with gene expression. In fact, only in 18% when we take matched samples, it's correlated very tightly. So there is 82% of the methylation that is not directly correlated to gene expression. And I think the last part of your question is it can actually go both ways. So the methylation, the 18% of methylation can affect gene expression, but it could also be a result of gene expression. That depends on whether a methylation marker is in the promoter, where it's more likely to affect gene expression. If it's in the coding region of a gene, it's more likely to be the result of gene expression. So I packaged the whole textbook into 2 sentences for you here.
And last question, Jason.
Thanks, Mike. First of all, I'd like to start asking my best friend, because he's dead, like I'm 4 years old.
Well, epigenetically 30.
Leong, genetics maybe these. Other process. We are when we change, looking to change to how we start that lazy. We're looking at years, months, days.
Oof. Yeah, now you get me sweating. So there were some papers that suggest very rapid methylation changes. I think when we talk, and this goes back to the question about lifestyle-induced changes versus pharmacologically induced changes, where the latter might be quicker. So, for example, vitamin C is actually sort of measuring, changing the epigenome to some extent because it regulates TET11, which is an enzyme that removes methylation. That can be, there were papers that showed it can be in minutes after exposure of cell cultures. I think, if you look at some of these trials that we have been involved in, they are clearly over more like months and so forth. But, it probably depends on the trigger for the change of methylation. It probably depends on the tissue, whether you have a rapidly dividing tissue versus a tissue that doesn't divide and all kinds of things. So there's also been more recent work where people use CRISPR to change some of the methylation sites and see to the extent that they then affect say, the functioning of the clock and things. So it's a very active field, and I think this jury is still very much out. It's of course not easy to do, especially not in living humans.
Okay. Thank you very much, Mike. And Mike is going to stay. He's going to be one of our judges too for the trainee presentations. And now we're going to go to the next section, and I'm going to have John Asser from the School of Pharmacy and Pharmaceutical Sciences chair the next session that is dedicated on Carlo Pasuk.